Hello, students. Hope you all will be fine. Let's start chapter number 4, Cell Cycle, part 2. In this part, we will read about the meiosis. Meiosis is a type of cell division which gives rise to four daughter cells, each having half the number of chromosomes of the parent cell. It takes place only in cells involved in sexual reproduction. Each parent cells are usually diploid, which by this reduction division produce haploid daughter cells. Cells which have two sets of chromosomes are called diploid, and cells with half the number of chromosomes are called haploid. Chromosomes in a diploid cells are in homologous pairs. Haploid cells carry one member of each homologous pair. Meiosis is a continuous process, but for convenience is divided into interphase 1, meiosis 1, interphase 2, and meiosis 2. In interphase 1, which is before the onset of division process, cells forms a copy of its genome by replication process. That is why chromosomes appear with two chromatids during division process. Meiosis 1 is a reduction division which for daughter cells reduces the number of chromosomes to half. Firstly, karyokinesis forms two haploid nuclei, which is followed by cytokinesis. Karyokinesis in meiosis 1 is further divided into prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 is the longest phase of meiosis. In prophase 1, crossing over occurs, in which the chromosome exchange genetic material, increasing genetic variation. Chromosomes condense and become visible. Homologous chromosomes, same size and genes chromosome, pair up. This is called synapses. Nuclear membrane breaks down and spindle fibers form. Let's have a look on the process of crossing over. Homologous chromosomes line up point to point against each other and form pairs. Their centromeres are in the same position. This pairing process of homologous chromosome is called synapsis. Each pair is called as bivalent. The non-sister chromatids of homologous chromosomes join each other at some points along their length. Such points are called as chiasmata. Each chiasma is a site for the exchange of some genetic material between chromatids. The exchange of segments of non-sister chromatids during prophase 1 is called the crossing over. In metaphase 1, bivalents are arranged at the equatorial plate. Spindle fibers are attached to the centromere of chromosome. One member of a homologous pair receives a spindle fiber from one pole and the other member from the opposite pole. Anaphase 1 has the spindle fibers pull on the homologous chromosomes. Separated chromosomes of each pair move to the opposite poles. Each pole receives haploid set of chromosomes. Once at their pole, telophase start, in which chromosomes uncoil into chromatin. A nuclear envelope is formed at each pole around the haploid cell. And then cytokinesis occur by cleavage in animal cell by forming cell wall in the plant cell. Although each daughter cell is haploid with half the number of chromosomes, their chromosomes are composed of two chromatids. Due to crossing over, these chromatids are not genetically identical. They must be separated in the second meiosis. So, two haploid daughter cells thus formed enter in interphase 2. And in interphase 2, the S phase is missing because we don't have to duplicate the DNA. As I described earlier, that there is no further DNA replication during the interphase 2. So the cell entered into meiosis 2. It is similar to mitosis and is divided into prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 2, chromatin coils again, so chromosomes appear as thick fibers, spindle apparatus is organized, nucleoli disappear, and nuclear envelope disintegrates. In metaphase 2, chromosomes line up separately around the equator of the spindles. Spindle fibers are attached to the centromere of chromosome. Like mitosis, here each chromosome is attached by a fiber from both poles. In anaphase 2, centromeres divide and spindle fibers pull the chromatids to the opposite poles. In telophase 2, chromatids reach at their poles and uncoil. The spindle apparatus disappears and nuclear envelopes are formed around the chromatid at each pole. In cytokinesis, cleavage furrow in animal cells and cell wall formation in plant cells give four daughter cells at the end of meiosis process. All cells formed by meiosis process are haploid as they have half number of parental chromosomes. Also, they have changed genetic makeup because of the crossing over between homologous chromosomes during meiosis 1. So this is the summary of meiosis. So in prophase 1, homologous chromosomes pair up and crossing over occur. Metaphase 1, homologous pairs line up in the middle. Anaphase 1, homologous chromosomes separate. And telophase 1, two haploid cells form. In prophase 2, new spindles form in both haploid cells. Metaphase 2, chromosome line up again. Anaphase 2, sister chromatids separate. And telophase 2, in which four haploid cells are formed. Let's have a look on the significance of meiosis. Meiosis help in the maintenance of chromosomes. During the life cycle of sexually reproducing organisms, diploid germ cells undergo meiosis to produce haploid daughter cells, which act as gametes. Fusion of such haploid gametes thus maintains chromosome number specific for each species. Humans have 46 chromosomes in their somatic cells. The haploid gametes, eggs, and sperms formed by meiosis have 23 chromosomes. In fertilization process, a 23 chromosome sperm fuses with an egg also having 23 chromosomes. The original chromosome number of 46 is restored in the zygote. In plant, meiosis takes place during spore formation. Meiosis also help to create genetic variations. Variations are the differences among the members of same species. They are necessary for the survival of species in always changing environment. It is because of meiosis that new combinations of genes appears in gametes and then in zygote. Crossing over and independently formed combination of chromosomes in gametes are two important events of meiosis. Both crossing over and chromosomal combination provide basis of variations. Meiosis is also essential for sexual reproduction because it produces gametes, egg and sperm in animals and spores in plants. Without meiosis, sexual reproduction would not be possible. Let's compare the two processes, mitosis and meiosis. Mitosis occurs in somatic cells. It divides only once. It produces two daughter cells. Daughter cells produces are diploid. Daughter cells become part of somatic body. All chromosomes remain independent of each other. No chiasmata formation. There is no crossing over during mitosis process. Daughter cells are genetically alike. In most cases, daughter cells can divide again, but in meiosis occurs in germ-like cells. It divides twice. Four daughter cells are produced. All daughter cells are haploid. Daughter cells form gametes. Homologous chromosomes pair with each other. Chiasmata are formed. Crossing over takes place during meiosis 1. Daughter cells are genetically different from each other and parent cell. Gametes produced by meiosis cannot divide anymore. That's the end of the chapter number 4. Hope you understood the concept. Thanks for watching. Like the video and subscribe the channel for more videos.